Video, please.
IICA welcomes you to this event and thanks you for your participation. To improve the quality of the video conference, please take note of the following recommendations. Remember to unmute your microphone only when you are given the floor. Please keep your microphone muted when another person is speaking. We ask that you keep your camera on at all times so that the other participants can see you. If you need to step away for a moment, you can turn the camera off and turn it back on when you return. Please put your cell phone in silent mode to avoid any interruptions during the video conference. We recommend that you maintain proper posture and position yourself adequately in front of your computer. Simultaneous interpretation will be provided for this meeting via the Zoom platform. At the bottom of the Zoom screen, you will see a globe icon that says Interpretation. Click the icon and select the language you wish to listen to. It is very important that you select the language in which you will speak, as this will facilitate the interpretation. At the end of the video, please remain silent for 30 seconds. Afterwards, the moderator will begin the meeting. Thank you very much for your attention.
Welcome back, everybody, to this day one of a 3-day workshop on the afternoon session. Welcome back, everyone online. I'm pointing to the screen. I can't see you, but you're somewhere in the clouds. So we have 5 sessions normally this afternoon, and we'll have about a half-hour set of Q&A at the end. I'd like to start with Dr. Signori from the CFIA, and I will introduce the following speaker, and then I'll ask my colleague Susana Flores to introduce the next 2 speakers after that. Over to you, Dr. Signori.
Okay, well, thank you very much. Just want to reconfirm that everybody can see my slides and hear me well enough.
Yes.
Excellent, thank you very much. Okay, so thank you for that introduction, and I want to extend a very gracious thank you to the organizers of this event for giving me the opportunity to present some of my work here today. I'm going to take this opportunity to tell you a little bit about how we use genomics and bioinformatics to gain insight to the evolution, the host dynamics, and the spread of H5N1 avian influenza viruses across Canada and across the broader Americas. So I want to just very briefly start off by giving you a history that tells you how we got to our current situation. So we know that avian influenza viruses can be broadly split into 2 groups. Lowly pathogenic viruses, meaning they are generally survivable to gallinaceous birds, primarily chickens, and highly pathogenic viruses, meaning that they're not. So lowly pathogenic viruses can become highly pathogenic if they are of the H5 or H7 subtypes, and they spontaneously have the insertion of a few basic amino acids at the HA cleavage site. So the hemagglutinin protein needs to be cleaved for proper function. In lowly pathogenic viruses, that cleavage is done by host proteases. And in the lowly pathogenic viruses, only host proteases that are localized to the respiratory and gastrointestinal tracts can cleave that protein. And so that leads to localized infections. However, in the highly pathogenic version, with the insertion of this multi-basic cleavage site, this protein can now be cleaved by proteases that are ubiquitous throughout the system. This leads to a systemic infection, leading to highly pathogenic infections. And so this typically happens in poultry, and which can then spill over into wildlife. And so this causes severe, but generally localized outbreaks of highly pathogenic avian influenza. And the opposite can happen too. It can emerge in wildlife and spill over into poultry, but this is much more rare. And so because these viruses are highly pathogenic, and because sick birds generally don't travel very well, traditionally the spread of highly pathogenic avian influenza viruses has been limited. However, that has been changing. So this here is the phylogenetic tree that basically summarizes the history of H5 influenza viruses. In 1996, a new group of viruses emerged that were highly pathogenic, but unusually persistent in wild birds, primarily wild waterfowl and shorebirds, so anseriforms and quadriforms. And that persistence in wild birds increased and increased. So the pathogenicity in these two groups of birds, migratory waterfowl and shorebirds, were reduced while this virus was still highly pathogenic to others. So culminating in this H5 clade 2344b that is now the underlying cause of the global outbreak. And so the dynamics of the virus globally have changed. We went from a situation where lowly pathogenic viruses circulate, occasionally spill over into birds, and become highly pathogenic, causing severe, but again, localized outbreaks. The situation now with 2344b is that we have the global spread of highly pathogenic viruses because it travels quite well in migratory waterfowl. And if we take that same tree, we can see this happening historically if we recolor the tree according to location. So we can see that in the early 2000s, this virus was entirely localized to China. However, as the virus became less pathogenic to migratory waterfowl, it expanded its host range. It went from China to Africa to Europe and eventually to North America. And this is summarized quite nicely by a recent paper by Xia et al. So what this graph represents is the relative time the virus has spent in a given location. So the orangish brown color here represents China, and we can see that prior to 2013 and shortly thereafter, the virus was basically entirely isolated to China. But as it got better at being carried by migratory waterfowls, that presence spread from China to Africa to Europe. And now we see that it looks like Europe is actually the epicenter of this clade of viruses, which is troublesome for us because Western Europe is just one migration away from North America via the East Atlantic Flyway. So with that, with the epicenter of this virus moving towards Europe, shortly thereafter, the virus was detected in North America. It was first detected in a blackback gull in St. John's, Newfoundland. This gull was technically collected in November 2021, but it wasn't determined to be infected with H5N1 until mid-December. This virus actually came over here again on the West Coast from Japan shortly thereafter, and it's come over again from Japan and again from Europe and again from Japan. So it's actually coming over during multiple migratory seasons from both coasts. And these are just the detections of reemergences in Canada. If we look at studies done by the USDA in Alaska, we actually see that it's coming over there from Japan and Korea quite often as well. But what's interesting is that the majority of these incursions from either Europe or Eastern Asia have been of little consequence to the overall spread of the virus across the Americas. 98% of all the viruses that we collect in Canada can trace their ancestry back to this initial incursion in November 2021 in a blackback gull. And we know this thanks to an enormous surveillance effort. So Oliver touched on this briefly, and I just want to take a moment to acknowledge them again. Canada's interagency surveillance program for avian influenza viruses. To date, they have collected and sampled almost 35,000 live, dead, and sick animals, the majority of which being anseriforms. As we know, they are the primary reservoir of avian influenza viruses, but also quite a lot of sampling of shorebirds, passerines, raptors, as well as terrestrial and aquatic mammals. Now, of all this sampling, about 10% of the samples come back positive for HPAI. On the domestic bird side of things, at the Canadian Food Inspection Agency, we have a national emergency operations centers for HPAI, and they have been quite busy. So since the incursion in November 2021, over 400 farms have been infected with high-path avian influenza, which has impacted 11 million poultry just in Canada alone. And as we heard earlier, that number is almost over 90 million in the US. So that's a lot of damage done by just a lipid bilayer with 8 segments of RNA in it. So most of that RNA, at least collected in Canada, comes back to the National Center for Foreign Animal Disease, where Oliver detailed how they've been putting in a lot of effort into developing and optimizing tools to quickly and accurately sequence the genomes of these virus. And so since the incursion, we've now collected thousands of viral genomes. And with those, we can gain insight into how the virus has evolved, how it spreads between different host groups, and how it spreads geographically across Canada, the US, and beyond. And so you're likely familiar with how one would assess the evolution of a virus with genomic data. Phylogenetics is not a new tool, it's been around for decades, but you may not be quite as familiar as how we would understand host dynamics or the geographic spread based solely on genomics. So I want to take just a tiny bit of time just to show you how that's done. If we take a basic phylogenetic tree here, where the tree branches are scaled by the number of mutations, so this shows the relatedness of a group of different genomes based on the distance and number of mutations between them all. If you've been fortunate enough to have your surveillance team collect a suite of epidemiological data, starting with dates, we can apply that to this tree to estimate divergence times between different groups of viruses. We can do this because we can make a regression of mutations over a collection date, which gives you a roughly linear relationship and allows you to estimate the number of nucleotide substitutions that happen in a given year. And so if you know substitutions per year and you know the substitutions on a branch, you can rescale the tree to go from a divergence tree to a time-scale tree. And so with a time-scale tree, let's say we want to estimate the divergence or the emergence of this group of viruses here. This group emerged in mid-September 2022. But we can take this a step further. We can add additional metadata to this tree. And using the shape of the timescale tree and the relationships between different viruses, we can infer epidemiological processes. So this is called phylogenetic. If we know the viral host information for each tip, we can apply them here. So we'll just simply apply domestic or wild birds. We can do an ancestral reconstruction of discrete character states based on the topology and the branch lengths of the tree to infer whether the ancestral nodes at all parts of the tree were either derived from a wild bird or a domestic bird. And that allows us to infer viral spillover from wildlife to poultry and vice versa. So if we look right here, you see the tree transitioning from gold to blue, meaning that is an inferred spillover from wildlife to poultry. We see the opposite here, where the tree transitions from blue to gold, inferring a spillover from poultry to wildlife. And then in this group up here, these actually represent 3 different farms. So this is actually inferring poultry to poultry spillover between different farms. So we can take this a step further and infer other discrete characters. We can essentially infer any discrete character we choose, including farm. So the tree has now been reconstructed to have the ancestral farm done at each node. And we can infer farm-to-farm transition using phylogenetics and a little bit of epidemiological data collection. We can also use this to assess geographic spread all the time, because obviously you would know the locations of these farms, so these can be mapped. And you can generate a plot of how the virus is spread over time. This plot is, of course, this data is made up, so this isn't real, it's just for demonstration purposes, but you can see what I mean. So these discrete character reconstructions are great when your samples are binned into discrete locations like a farm. They don't always work with wildlife because they can't really be confined. We can do it with larger regions, but this gives poor spatial resolution. So if you want to assess the spread between Canadian provinces, there's only 10, and it doesn't give you very good resolution as how the virus spreads. You can add many smaller regions to the analysis, but this requires too much computation. So there's another way that I'm going to show you and give you some nice examples of later of a continuous character reconstruction. It's essentially the same, but instead of reconstructing discrete character states, we reconstruct numerical values. And this gives you a nice smooth transition across the tree of whichever numerical trait you've collected. And for our purposes, we have latitudes and longitudes, which are both location and numerical. So we can end up with something like this. I hope you can all see the video nicely. This is a reconstruction, a continuous reconstruction of the spread of the whole Eurasian virus that came here. It was first detected here in November 2021. So it came over from Europe. It got here just in time to catch a ride south on the winter migration down south. In the following spring, it worked its way back up through the Midwestern United States and then emerged in Western Canada. What's interesting about this particular reconstruction is not necessarily where it spread, but where it didn't. Each of these dots represents a collection, and you can see there are relatively few as you go further north into Western Canada. And this is actually a bit strange. I can get to the next slide. So this is just a static version of the same reconstruction. The virus essentially stopped in the Midwest United States. It went down and then up and then stopped. And where it stopped is interesting because it stopped right on the doorstep of what's called the prairie pothole region. The prairie pothole region is one of the most prolific waterfowl breeding areas on the planet. It supports about 50% of North America's migratory waterfowl. So it's interesting that a virus that spreads by migratory waterfowl didn't spread through a region, one of those prolific migratory waterfowl breeding areas. Well, as it turns out, this particular genotype didn't spread through that region because what happened was this virus reassorted and gave rise to new viral genotypes that replaced its ancestor. So just to review, reassortment is when two viruses co-infect the same host cell. During the genome replication process, their genome segments get jumbled up and then repackaged into new combinations, leading to new viral genotypes. This has happened a lot since the virus came here. So if this singular blue dot represents that Eurasian virus that was first detected on Canada's East Coast in November 21, it reassorted and reassorted and reassorted to where in the 18 months that followed, there are 37 distinct genotypes. These particular dots are scaled according to how frequently these genotypes are collected in Canada. You can see that most of them are relatively inconsequential. They're very small collection numbers. There are 6 major ones. And so if you are familiar with the USDA's GenoFlu genotyping program, I've put those genotypes in brackets next to our internal numbers just to make things easier for you if that's what you are familiar with. So what's interesting to note about all these viruses is that despite all of these being H5N1 viruses that can all trace their ancestry back just a few years to November 2001, they seem to behave differently. They have different geographic spreads, and they have different host dynamics and different pathogenicity. So I just want to give you a quick example. These two viruses here, one evolved from the other. They occupy essentially identical geographic regions. So they had access, these viruses had access to the exact same animals. But if we look at the host dynamics, they're actually a little bit different. So what these spiderweb sort of plots represent is the thickness of the lines represent the frequency of viral transmissions from one host group to another. We can see that the virus on the bottom, the derived state virus, the frequencies are heavily biased to go from anseriforms to other host groups, while as the genotype on the top, it's a little bit more balanced. We have laboratory data for each of these viruses, and it can kind of help explain this for us. If we look at ducks infected with this virus at the bottom, they shed way more virus than they do when they're infected with others. So this might explain why this virus is so prolific at being spread by anseriforms. There's another sort of oddity with these host dynamics in that the virus on the bottom is, by and large part, almost exclusively, if we exclude domestic transitions, the transmissions from anseriforms to other wildlife is almost exclusively going from raptors to mammals. So these are two groups that aren't, relatively speaking, aren't commonly infected with avian influenza. But again, if we look to our laboratory data to try and interpret this, we see that this virus on the bottom is actually the least survivable virus that we've ever tested. So it makes sense that it's being spread from anseriforms primarily to two species that are largely eating carrion. But as you can see from the maps that I'm showing you here, these two viruses had very little influence on the broader spread of H5N1 across the Americas, but rather that was the result of these 3 genotypes here. So I've already showed you the reconstruction of the whole Eurasian genotype, what the USDA would call A1. It spread down south on the Atlantic seaboard and then back up to the prairie pothole region in the spring, where it stopped because it reassorted to create another genotype that replaced it in this region and spread like crazy throughout the prairie pothole region, but it didn't spread much else after that. You can see its geographic spread is essentially limited to the prairie pothole region because what happened, this virus reassorted to create another virus that replaced it in the prairie pothole region and then spread more broadly across Canada and the United States and beyond. So what we can see from these maps, and if we look at how frequently these viruses are collected over time, they appear to be replacing each other. A virus spreads, stops, reassorts, and gets replaced by its descendant over and over and over again. They appear to be getting more and more fit as time goes on. And so if we estimate the relative fitness of these viruses, that's exactly what we see. We see successive reassortment events giving rise to a more fit virus, culminating in this virus here. This is the most frequently collected virus in Canada and the US, and it has the highest fitness index as indicated by that figure we just saw. This virus, like I said, it replaced its ancestor in the prairie pothole region. And then, as it was popular in this region, in the following migration season spread not just north-south, but also east-west across the United States and then further south as well. It was able to do this because this prairie pothole region, although it is in the center, essentially, of Canada and the US, it overlaps, at least in part, all 4 North American migratory flyways. So birds from different longitudinal flyways mix in this region, and that allows the virus to spread east-west in addition to spreading it north-south. And if we look even further south, from the genomes available on GISAID, 92% of them from Mexico, Central, and South America are this particular genotype. And in fact, it is this genotype that has made its way down to Antarctica. So for the people and the penguins of the Southern Hemisphere, this surveillance in the prairie pothole region can have some predictive power. Surveillance in the spring and in the fall can tell you which genotypes might be popular in overwintering locations in the future, which is not an easy question to answer, because as I said, in the 18 months that followed the initial incursion of this virus, it diversified into 37 different genotypes. This was just the first 18 months. And if we update this to more recent times, that number has increased to 56. And as we can see, there's one genotype that I want to highlight here, a new fairly common genotype that emerged from what was the fittest genotype that we've seen. It took 6 segments from that, segments 3 to 8, and took 2 segments from a lowly pathogenic North American derived virus to create a new genotype, one that the USDA calls B3.6. What's notable about this is that not this virus, but this is the ancestor of the virus that is now infecting dairy cows. And we can get some insight and see why that may have happened, because if you look at how frequently these genotypes have been collected, these are just numbers in Canada, but we can see that this new genotype, B3.6, is by far the most commonly collected genotype across the Americas. And if we redo our fitness index analyses, we see that this is by far and away has the highest fitness index of anything else we tested, which is not surprising because it is by far and away the most commonly collected one. So what we're seeing is this is now the 4th iteration where a virus has reassorted and given rise to essentially a more fit version of itself. There is successive reassortments giving rise to more fit viruses. The virus is still constantly evolving into new genotypes as it reassorts with North American viruses. And as you can see from these collection data in Canada, the virus has been consistently collected. There are ups and downs, but it has been consistently collected since the outbreak began. So whether or not we want to declare it yet, but it seems like if it is not already endemic to the Americas, it's well on its way to becoming so. So with that, I need to thank an enormous number of people. All of my co-authors that are part of this study, once again, Canada's Interagency Surveillance Program for Avian Influenza, my colleagues at the NCFAD Avian Diseases Unit, our Genomics Unit, and then as well as the National Emergency Operations Center for HPAI. I want to thank all of you for your attention, and I would welcome any questions. I have built some time in for questions, but if the moderators would prefer to wait till later, we can do that too. Thank you.
Sincere thanks, Dr. Zoonotic. We're going to keep our questions till the end of the 5 presentations, and we'll have about a half an hour Q&A. And I really appreciated all the visuals. I think the room reacted really well to that. It's a good way to express the phylogenetics. And I wanted to now invite Dr. Tavis Anderson from the USDA Agriculture Research Service. Over to you, sir.
Good afternoon, everybody. Ideally, you are now seeing full screen on your side. Is that correct?
Yes, now it's in presentation. Thank you.
Fantastic. Thank you very much. Great. So I'm going to take a little bit of a slightly different trajectory from the avian talk that we just saw. Thank you very much. That was great. And focus in on influenza in swine. And tell a little bit of a story about some of the tools we're developing to analyze influenza A virus in swine and what the diversity in this animal means for human health and public health. And in pigs, there are three major subtypes that co-circulate endemically: the H1N1s, H1N2s, and H3N2s. Now, that's a little bit deceptive because there's significant diversity within those subtypes, and that's because of two major mechanisms. The first of which is the antigenic drift process, because the surface proteins are the target of neutralizing antibodies. And the second is, as you've just seen from the avian presentation, the process of reassortment and the emergence of new subtypes because of antigenic shift. Now, the way that we're able to track this and think about it is through a national surveillance program in the United States. So the first question I want to address is what's going on in the US? And the first thing that comes out of that is that we've now had about 15 years of data collection within the National Animal Health Laboratory Network. And this was implemented in 2009, though the construction and vision for this program was a little bit earlier as well. It's implemented through 40 different labs across the US, and it's a passive surveillance system, which means that pigs that are clinically sick or that have influenza-like illness are swabbed or tested, and then those swabs come into the National Veterinary Services laboratories or regional labs, and they're sequenced. Between 2010 and 2023, a subset were whole genome sequenced, but from 2023 onwards, all submissions into the surveillance system are whole genome sequenced. And we do have around about 11,000 strains that are available for research purposes in a national repository, virus repository as well. What comes out of that is that there's constant circulation of different H1 genetic clades. And so these are just a time series of the last 2 years of data, with the different colors in the center here highlighting the different genetic clades. And ideally, what you notice is that there are 9 different colors for the H1s. And each of these is genetically distinct and also antigenically distinct. If we flip to the H3s, we see a similar pattern, just with slightly different colors this time. Hopefully they're different colors. Again, with 9 distinct H3 HA clades. So within a 2-year window, in pigs in the United States, we have 18 different HA clades. Now, sort of for context here, when you go and get your human seasonal vaccine for influenza, you're getting at most 4 different components. And so here we've got 18 different viruses circulating in our pigs. When we flip more broadly to what's happening with influenza in swine in other countries, we've been fortunate in this case to work within a larger network called the OFLU network. And this is a group of swine influenza experts. There's also an avian influenza group as well, but this is the swine group. And we work every 6 months as part of this, or nested within this WHO biannual vaccine consultation meeting. And within those WHO influenza vaccine consultation meetings, there is a pandemic preparedness discussion that focuses on animal influenzas. Right now, within the OFLU network, we have about 12 to 15 countries that are sharing data and isolates. And this is centralized at the USDA in the National Animal Disease Center, and then also with our colleagues in the UK at the Animal and Plant Health Agency. So every 6 months, a whole lot of unpublished data, including from CFIA in Canada, is coming in into our analysis pipeline here. And what comes out of that is this type of realization that we've got 18 different HAs in the United States, and over the last 6 months, another 17 different HAs globally. So we're up to over 30 distinct hemagglutinin genes that are circulating in pigs across the globe, which is a rather large amount. And the reason why all of that diversity is troublesome is because some of those viruses then end up in humans. And this is a relatively regular event. And so now we're plotting just for essentially June 2023 to January 2024, all of the detections of swine influenzas that have been popped up in humans, and those are annotated with the orange crosses on the maps that you can see here. So we're getting variant cases in the United States, in Brazil, and also through Europe. And this is just the H1s. So we had eight H1 variant infections from 4 different H1 clades, and we also had another H3 variant that was detected, but we weren't able to get a genetic sequence on that specific case. So we're unsure of the clade. So essentially, there's regular zoonotic events from these swine influenzas that are out there. And they're not just one specific clade, they're coming from a subset of those clades. And we're still trying to work out why. And just to sort of go into that question a bit more detail now. So why is all of that diversity a problem? Why is swine influenza diversity a problem? And I think that the first thing to hopefully drive home is that I'm calling it swine influenza, and I like thinking about influenza in pigs, but most of our influenza are derived from human seasonal influenzas. And this is a relatively recent study that we conducted, and fortunately I don't have to go over phylogenetics because that was just covered. But what you're looking at now is a phylogeny, a time-scaled phylogeny of around about 15,000 sequences. The human sequences are colored black. The swine sequences are colored orange. The length of the branch reflects the duration of time in which these viruses were circulating in pig populations. And what you're looking at there, if we count across this entire phylogeny, is nearly 400 human to swine transmission events from 2009 to present. And this is just within the H1 pandemics. So one specific clade of those 30-odd clades that we have. The flip side is that there's 18 swine to human transmissions across the same time period from this clade. So almost all of these viruses that we're detecting in pigs have an ancestor that was a human seasonal influenza virus. Okay. Once it gets in there, what it's essentially doing is constantly recharging the evolutionary battery. So you've got new viruses coming into the pigs. Now, so this is just some machine learning modeling to basically split out the source of all of the viruses in the USA pigs. And on the right-hand table, you can see here the different human influenza seasons from 2010 up to the sort of 2019 season, and is when we stopped analyzing these data. And you can see the different number of spillovers here. So essentially, there's on average, 20 to 50 spillovers in a given flu season, and about 25% of those persist in pigs for longer than a year. And once they get in pigs, what they're doing is contributing to genomic diversity. And the way that they're contributing to genomic diversity is the next section that I want to talk about, is what happens when these human viruses get into the pigs. And there's 2 different things that I want to point out here. And the first of which is when you get a human virus into a pig, it can be dramatic. They reassort, and it can be a clade-creating process, so essentially speciation. And what you're looking at here is a neuraminidase phylogeny, the N1 gene in our swine influenzas, and we're mapping the reassortment events along this backbone of the neuraminidase gene. And I'm pulling out 3 distinct speciation events that are labeled here with the lightning bolts. And that was where there was a reassortment between a human seasonal virus and a pig virus, and then that spawned this massive diversity that jumped out of it. Now, with a whole lot of downstream detections of that specific virus as well that had that unique combination of genes. And this is relatively dramatic, but it tends to not always be the case. What we generally see is this next example, a sort of subtle shaping of the evolutionary landscape. And now we're mapping reassortment onto the N2 phylogeny. So this is 2 distinct N2 phylogenies on the left and the right that are just different lineages that are circulating in our pigs. You can see on the left-hand phylogeny, ideally just 1 black arrow. And that 1 black arrow represents a single reassortment event that kick-started a new virus that was transmitting in pigs. And on the right-hand side, you can see another 8 reassortment events. And this is going back 20 years. So over 20 years, there were only 8 reassortment events in this gene that were persisting in more than 10 subsequent detections. So reassortment isn't always dramatic. It can sort of just like happen occasionally and go away in the background there. And when it's going sort of bubbling along in the background, what's happening is epistatic changes in diversity. And on the left-hand side now, what you're looking at on this slide is sort of prior to that arrow, you're seeing two different evolutionary trajectories of the hemagglutinin gene and the neuraminidase gene. Then in around about 2013, there was reassortment, and that pairing then started to sync up. As you can see on these two lines here, there's just a strong correlation between the evolutionary dynamics of these two genes. And that was occurring because pre-reassortment, the viruses were just evolving and accruing mutations. Post-reassortment, there was a strong selective pressure with this new combination, and there was a rapid sort of fixation almost of a handful of mutations in both of the genes, in the hemagglutinin and the neuraminidase. So you essentially see fixation of these particular amino acid signatures. At least in this case, we're visualizing the neuraminidase gene. And there's only a handful of changes that are going on, but they have a dramatic impact. And so when we start doing sort of molecular modeling here, and we're looking at this is now back onto the hemagglutinin, when we're sort of looking at the hemagglutinin protein and those individual amino acid positions, what we start noticing is a handful of mutations can result in dramatically different protein structures. And so this is just 3 distinct clades that we pulled out and modeled. The colors here are representing individual different amino acid differences between those different clades. And this is just a crude way of comparing the protein structures between them. And you can start seeing that clade 1 viruses are very different from clade 2 and clade 3 and so on. So just a handful of small changes driven by reassortment can result in meaningful structural changes. Now, what does that mean for vaccine and pandemic preparedness? It means trouble sometimes. And so we can take those viruses and test them against human seasonal vaccines. And that's this graph that you're looking at now. So each of these broad columns represents a different H1 pandemic vaccine, from the California 2009 vaccine to a more recent Hawaii 2019 vaccine. Each of the colors on this graph represent one representative strain from each of the different spillovers. So in the top left here, you can see this sort of red color, an Indiana virus collected in 2021 that had been evolving in pigs for 7 years. It was a 2013-14 flu season spillover, all the way up to this Missouri virus on the far right that spilled over in 2019 and is represented by this star shape. Now, when you test this in hemagglutination inhibition assays, what you start noticing is that there's a smattering of color across the graph. And that's because each of the viruses is behaving and reacting in a little bit differently in this assay. But the sort of biological threshold we impose here is a twofold loss in cross-reactivity, this 2 value here. And essentially, every single swine virus that we test has at least one of these vaccines in which it has significantly reduced cross-reactivity. So if one of those viruses were to jump into a human, there is no prior immunity to that specific virus, potentially, depending on the vaccination history of that individual. So how does it happen, and how fast is it going on? In general, this is happening really, really fast. And what we tend to see with all of our data is that we're getting constant spillovers from humans into pigs. Occasionally, those spillovers get into the pigs, persist, and once they're in pigs, they reassort. And as they reassort, there are these sort of epistatic changes in the surface proteins that drift away from the seasonal precursor that founded that population. And frequently, it's really just a handful of amino acids that are changing. That's all it takes to reduce antibody binding. And so you can see in these lower phylogenies here, these sort of stylized phylogenies, we're mapping a handful of amino acids. On the left-hand side here, you're seeing position 155 and 187, those 2 positions. And then within that clade, 2 swine to human transmission events. And similarly, on the right-hand side, you're looking at 3 amino acid differences and cases going from swine back into human. So it doesn't take much, because these are human viruses that have just been evolving in pigs for a year, sometimes 10 years, sometimes 100 years. But they're just human viruses that maintain all of those mammalian adaptation markers. They're evolving and transmitting in pigs. So the next question then is, because that's a little bit of a bleak statement that the human vaccines aren't great against swine viruses, but is there anything else? Is there anything else that we can do that sort of can help prevent sort of another pandemic? And the answer is yes. And during those WHO meetings that I mentioned, there is this discussion about what's out there in pigs. Is it different from the human seasonal vaccines? Is it different from these things called candidate vaccine viruses? And these candidate vaccine viruses are regularly assessed within these OFFLU reports and WHO protocols. And this is a case in which we were able to detect a virus that was circulating in the United Kingdom, in England specifically. And we were able to compare that virus against a panel of other human seasonal vaccines and candidate vaccine viruses. There was no cross-reactivity, and it's a major virus circulating in England, and it had caused a variant case in 2023. And so this single strain then was recommended as a new candidate vaccine virus for pandemic preparedness, which is really encouraging because generally these candidate vaccine viruses, when they're available, they work rather well. And so this is just a subset of the data that we've got from one of our H1 lineages. All of the different colors on the branches reflect the different genetic clades that we're sampling from. You can start seeing the orange reflects variant cases, so swine viruses that have then been detected in people. And then the red reflects either a human seasonal vaccine or a candidate vaccine virus. And so you can see here, ideally, you can see blocks of red in the top, and then some candidate vaccine viruses down here that have been developed to control and prevent any sort of future pandemic. So where they're around, they're great. But they are challenged by how fast swine influenzas evolve. And now this is a larger panel, hemagglutination inhibition panel, where each of the columns represents a candidate vaccine virus or a human seasonal vaccine. The rows here represent different representative viruses circulating in swine. And then the colors represent the cross-reactivity between those swine viruses and the vaccine. And the bold boxes represent sort of the homologous reactions, the within clade reactions that we really care about. And what you can see in this top left color, the top left corner ideally, is that when we test our swine viruses against available vaccines and candidate vaccine viruses, we see this red color coming out of it, which is a greater than 8-fold reduction in cross-reactivity. And essentially, we wouldn't expect there to be any protection from that vaccine against this specific swine virus. Now, that is concerning because we also take these swine viruses and we test them against a pooled human serum, and that's on the far right of this table. And you can see the same color, the bright red, sort of the danger warning sign here, in that this specific swine virus, there is no prior immunity in the human population that we're sampling here as well. This does vary depending on the different genetic clade, but we try and pair the sort of the genetic analysis with the hemagglutination inhibition analysis, with also testing with the human sera as well to see what sort of reactivity patterns are out there. Now, this is sort of where I wanted to get to, is sort of how can we work out where the gaps are and improve all of this and improve our vaccine composition. Because there's a lot of diversity, there's a lot of viruses out there. And I think you saw with the avian influenza situation, it's as complicated as our swine situation, maybe more so. So when you think about genomic surveillance, how can you distill all of those thousands and thousands and thousands of viruses that are being collected in sequenced down to a handful of representative viruses? Now, the way that I sort of visualize that, and our team does, is with balls of diversity. I think of just like soccer balls of amazing amounts of diversity, and what we're trying to find is the center of that soccer ball that represents that soccer ball, essentially. And this is a relatively new tool that we've just released called Parnas that does this objective selection of representative strains. It has two major sampling strategies, and this is all sort of based on an idea called the K-medioid problem on a tree. And on the left-hand side, we're visualizing the first representative sampling strategy. And when you have all of that genomic surveillance data, you can ask the program to choose the 3 best representatives. So you just put in, I want 3, because 3 are the only that you've got experimental facilities and you can only test 3 viruses. This will give you the 3 that are the best. Alternatively, you can flip the objective, and that's the right-hand part of the slide. And you can say, choose as many strains as necessary to cover all of the diversity that is out there. And so you set a threshold here, a radius, and you ask it to identify the best representative under this threshold. And so you may get 3 viruses, you may get 10, or you may get 20. And then you can sort of combine these two things together to optimize the number of viruses that you can test with an objective measure of how much diversity is covered. To give you an idea of how this works, it's essentially this. You have a massive phylogeny. We've tested this on upwards of 100,000 strains in a phylogeny, and you'll get an answer within 10 minutes or so. And what the algorithm will do is partition your phylogeny into diversity groups, into those soccer balls. And so now this phylogeny is colored by 3 different colors, and it will spit out 3 specific strains that represent those different colors. And then it will tell you how much of the diversity that is accounted for. In this case, for a human H1 pandemic situation, with 3 representatives, we're accounting for 44% of overall diversity in this phylogeny with 3 strains. So not a huge amount of diversity with 3, but objectively, we know that these are the best ones to choose. You can also start eyeballing how many strains are necessary to test. And what you start seeing here is, in general, you start seeing diminishing returns as you increase the number of strains that you're testing. So this is another objective way of identifying how much testing is necessary. When should you stop? You don't need to always test everything, though it is quite satisfying. Sometimes you can optimize and do 2 or 3, and you're still covering a fair amount of the diversity that's out there. In our case, we start seeing diminishing returns after 4 or 5 strains. So we can normally start optimizing what we want to test. We can also determine how long strains remain representative. So once you've got your laboratory strains and assays established, you can start asking, when do they need to be updated? And in this case, ideally what you see on the left-hand side is 2016 selections, 2017, and then 2018. The top panels represent H1s. The bottom panels represent H3s. And what we did here in this case was in 2016, we took 2016 data and made selections, either 2, 4, or 6 representative selections for the H1s, 1, 2, or 3 selections for the H3s. And if you do that, if you take 6 selections for H1s, you get 90% coverage in 2016. And if you hold those same selections static into 2017, you still maintain 90% coverage. But then if you hold those same selections again into 2018, you see an almost linear decline. And that sort of linear decline is relatively consistent across each of these different panels. So that we're starting to get this idea that every 2 years, we have to start updating the assays and the strains that we have in the lab because they're no longer representative of what is out there in the field. And this is a very broad picture, but you can imagine here that you can tweak this objective and you can identify it and use it for when something is unique. And that's the next slide that I want to show you, is you can use this framework to identify gaps in coverage and identify genetically novel genes. So what we're doing here is we were able to take some human seasonal influenza viruses, build a phylogeny, and then rescale it based upon its phenotype. So we were able to integrate the hemagglutination inhibition assay data, and then you can collapse all of the 2,000 strains into something that's about the same. So we use a 2 antigenic unit threshold. So all the flat lines are all basically viruses that are the same. It's tough to believe, but there are 2,000 viruses on this phylogeny. And when we do it this way, we can then say, given these prior vaccines, and in this case, it's the red square in the top left and the red star in the lower right are the 2 human seasonal vaccines. If we hold those static, we can then say, are there viruses out there circulating that are significantly different from other viruses? And are they significantly different to these vaccine strains that we're using? And in that case, what we found was a chunk of viruses right in the center here in this 2B labeled area of the phylogeny. And what that would suggest then is if we wanted to have complete coverage and complete protection, we would need to make 1 more vaccine selection right here. And those 3 selections then cover all of the 2,000 completely. And so The last component then is associated with that, and that's called evaluating prior selections against new genetic surveillance data. And this is, I think, one of the constant themes that I've seen as I've been sitting on the talks today, is that there's a wealth of data that are coming through genomic surveillance activities. But what we'd ideally be able to do is say, when the data are coming in, is this about the same or is it different? And so this tool gives you a way of evaluating your prior selections or your prior data against the new genetic surveillance data. And so this is just a simple command that you run in the tool here. And basically, it finds the best selections, or it finds your prior vaccines, or it finds your prior lab strains, and then compares your new data against those prior selections. And in this case, it's essentially saying that there's about a 12 to 13% difference between your new data and your old data, and you should probably update either your vaccines or what you're using in the lab. And so just to wrap up that whirlwind of swine influenzas, and I should have dropped in all of the cattle H5 stuff, but I decided to stick with swine, is that in swine, we've got 34 odd different hemagglutinin clades. And that's just in a 6-month window. And that's reflecting a relatively limited surveillance in a number of different countries. And from those 34 different hemagglutinin clades, we have human variants reported from 4 of them, along with another H3 variant. Just in 6 months, we've got 8 variant detections. So that's a relatively significant amount. The reason for why we're trying to work out what's going on in swine is that it's a challenge for public health. And that's because all of the viruses that we have in pigs in the US and in general globally reflect human to swine transmission events. So these are essentially human viruses that are just evolving and transmitting in pigs. All of the swine influenzas maintain human influenza genes, specifically the nucleoprotein and the matrix gene in particular, and they have features that are associated with or very similar to the H1N1 pandemic. Some of the gene constellations also have a significant impact on transmission efficiency, and in general, subtle differences between strains alter the phenotype and can result in zoonoses. So what we're trying to do is build in some objective tools that allow you to select HA genes for characterization, and you can flip the objective around in these tools to determine whether you have a good vaccine and address when you need to update vaccine components. All of the software that we build is publicly available, and you can find the GitHub page right there, or you can send me an email if you'd like some tutorials on how to use the tools. I'd like to acknowledge our team at the National Animal Disease Center, part of the Agricultural Research Service and USDA. And we receive funding from USDA ARS, APHIS colleagues, the NIH, and CDC. Thank you very much for your time today. I really appreciate the opportunity to present this work. Thanks.
Great. And thanks a million, Dr. Anderson. That was a very interesting talk. We will keep the questions for the end of the meeting. Ahora vamos a continuar con nuestro siguiente ponente, el doctor.
Now we're going to continue on with our next presenter, José Iván Betancourt from the University of the UNAM. Dr. Betancourt.
Gracias, doctora Susana, por la invitación.
Thank you, Dr. Susana, for the invitation. We're going to talk a little bit about what we have found in Mexico in the swine fever, swine influenza.
Okay.
Very well.
Muy bien. Bueno, pues,
Very well. Okay, so
el foro de hoy ha sido influenza.
today's forum has been influenza. In reality, you know
Que el cerdo
that the swine, that the pork, many times is associated as a mixture of the virus of influenza from origin of other species. And in being an illness that is multi-species, it becomes emerging, especially when we're talking about the variability of the viral subtypes that we can find in the animal kingdom. The next one, please. It is important then to have it very clear what part of pork plays and the different outbreaks that we have had for more than 100 years now. The first one of them was in 1918 with the Spanish flu by a subtype H1N1. But nevertheless, years later, it was isolated to the swine, to the pork. In 1957, the Asian flu, H2N2. In 68, the Hong Kong flu, or an H3N2. In 73, 77, the Russian flu, another type of H1N1. And in 2009, the Mexican pandemic, or an H1N1 type of origin, Eurasian origin, because it was not a virus that was completely human. It was not a virus that was completely porous or swine. It had been recombined. In the case of the pig, in 1970, we see the transmission into species because previously the human had previously been infected with the H3N2, and we can see this in 1968, 1967, 1978, and 98. There's a combination between the human virus report, H1N1 and H3N2. And we generate this subtype that is H1N2 that is prevalent in Canada, the United States, and later it's also reported in Mexico. In reality, in 2009, we started investigating about influenza. The great concern for everybody was precisely as the priority of the WHO is the N5N1 subtype of pathogenicity. Even before that, in 2006, there was already confirmation of wild birds that were affected with H5 and 1. It wasn't anything new. It was something that had been on the process for many years now. And you can see how here many of the continents have been reported with different types of birds in the H5N1 subtype. The next one, please. What happened with the humans? Because the previous slide was about wild birds. In humans, in 2009, this is a map that was published by the WHO, basically beginning with the pandemic outbreak. And you can see that there are some zones in which there is always reports of the aviar H5N1 in humans. And we had already reported some mortalities, like in Cambodia, Vietnam, and in other words, in many areas of the Asian continent. As a matter of fact, in 1996, the first case of the H5N1 of high pathogenicity in humans type took place precisely in Hong Kong. A virus that had a person that had a contact in a farm with different birds, poultry. They assumed that they were positive. He's sick, and after that, another 60 people get infected and they die. This is from 1996. This is a photograph of 2009 regarding the subtype that is most concerning to the sanitary part worldwide. Next one, please. Finally, we're talking about a virus that has a great capacity, a mutation capacity, because it is a virus that changes and also a recombination or resettlement. Other exposures have already mentioned it. 18 transfers have been reported in the other species, and we can even have up to 198 subviral subtypes in the animal kingdom. Of course, not all of them have been identified, but at least in the swine, the three most common subtypes are H1N1, H3N2, and the subtype H1N2. The next one, please. It is important to identify what type of hemagglutinin the virus had that affect the hogs, the pigs. As Dr. Macalister said, there are several H1N1s. In other words, it's the same subtype, but there are variations that determine if they are in different clusters. This is very important because this makes the variability even greater, apart from the virus subtypes. It is the responsible of the union of the virus to the cell This is merged with cell walls, the cell membranes, and it is a main gene to which the antibodies go by the immunological system. The next one, please. The second gene that is most studied is neuraminidase, that has the functionality of sialidase. Once the virus is repeated, there is a breaking of the links that maintain the virus together, and at the moment of coming out of the cell, it allows it to cut through and infect other neighboring cells. The next one, please. There's also a conversation going on, or we know that there are possible mutations in these genes, especially in the mountain. And if it falls into one of the 4 or 5 antigenic or immunogenic positions, this is what the mutation is going to generate is a change of a base for an amino acid. There is a mutation from a base, and it generates a new amino acid. If it were so, there would be some virus that are the same subtype, but they are pathogenically different because they have some changes of ethnicity associated to the change of amino acids. And on the other hand, it has been seen, and what I'm going to demonstrate is resettlements. In other words, different genes of 2 or 3 virus that are able to generate a new virus, a new virus subtype. The next one, please. This is the panel of the different class that we see, but nevertheless, if we focus on the top parts where we have the figure of the swine, of the pig, the subtype H1 and H3 are the ones that are most common, the most recurring, found in the porcine swine species. Nevertheless, the interaction that there is with the other species that they have around them, the ecological niches specifically, are going to generate the increase of the risk because it is an omnivore that is a receiver of possible virus from other species. In other words, we have in the photograph where we have the hogs drinking water and also some chickens drinking water for there. So those niches are of high risk for the generation of new recombinations of virus, of the influenza virus. Next one, please. Here we have a simulation of a cell and a virus that is identified for many different receptors. In the case of the swine fever, they have the cellite 2.3 that is in the cells, and also the 2.6. And that is why they have the capability of having the cells infected by human virus also, which are the ideal receivers for the alpha 2.6 virus. And in the case of the birds, the alpha 2.3. Next one, please. Very well. Nevertheless, there's an article that demonstrates that human beings also have receivers that are similar to those of the porks, although in a different concentration and higher versus a lower, sorry, I said a higher respiratory routes compared to low respiratory routes. And this is going to generate that the human being is also a vector of the influenza virus, and also swine fever, for swine fever. It has been demonstrated that humans have been infected directly from avian birds, but they have also seen human infections from a pork origin or swine origin. The typical clinical types of the influenza, the hogs are very well known, and the pigs are very well known. Nevertheless, apart from the respiratory problem, we have seen that some viral variants are more aggressive in pigs. And we're talking about viral variants in some of the cases when it is a new viral subtype, or when it's the same subtype with some mutations that make it more pathogenic. Next one, please. This slide is very interesting, and although the most common ones of the subtypes that is found in the hogs are H1N1, H3, and 1 and 1, 2. And there have been reports in the United Kingdom of a subtype H1N7 infecting hogs, H1N1 in Italy in 2009 affecting hogs or pigs, H3 and 8, which is normally for a queen and dogs, also affecting the hogs, which is the purple one there. In China, H9 and 2 in 2008. In the United States, H4 and N4 also affecting pigs. H3 and 3 in Canada in 2004. H4 and 6 in Canada 2000, and finally the subtype H2 and 3 in the United States also reported in Seattle. What does this mean? Well, it means that the pig is susceptible to being affected by virus from other species, and that fortunately, the majority of them has not adapted to the swine or to the pork uh, species that to have a pork-to-pork infection. That has not been the case, but there is a possibility. The next one, please. The first research that we did was a post-pandemic. We wanted to find the pandemic virus in hogs or in pigs, and we put together a route of diagnose. The sample was processed in a very with a S3 variety security, and we would do the extraction of the DNA, and we would do a real-time extraction. On the other side, we would go to the left to the PCR final point to see if that if the sample was positive, that we might be able to create the sequencing. All the positive samples we would put them into a chicken embryo to achieve a viral infection, to have the opportunity of having the virus, the obtention of the virus. And on the other hand, we also look for the PCR multiple in the sequencing. Next one, please. Out of the different states of the Mexican Republic, we obtained 1,210 samples. And the state where we obtained the biggest amount of samples was for Jalisco, from with 105, and then Querétaro, and so on, and in which we had access to things. The next one, please. All of them were processed by PCR in real time, and we only found 24 positive samples. Some of them we were able to do the sequencing. The next one, please. We did not find in those 1,210 samples the pandemic virus, which was the objective, to be honest. The pandemic virus, as you know, it comes from resettlement in which we have some genes that came from the duck. Later, that is mixed with two of its genes with the classic virus, swine virus, and two more, neuraminidase and protein N, that go from the duck to the pig in 1979, and that's why it has that Asian origin. Next one, please. What we did find, and it was very interesting, in some of these samples, is that although we did not find the pandemic virus, we found a virus that we noted to the Big Bang. It was identified as ACA in Mexico. It was a subtype of H1N1 that has in its nucleoprotein and in the polymerase amino acids that are preserved precisely to the virus of the pandemic, and that located as the ancestor or the origin of the pandemic virus. We know that the case in this case was in Mexico, but nevertheless we didn't have it very clear if there were among the virus that we had obtained and isolated any virus that was similar to the pandemic. Once we identified this and it was published, we did another study, the next one please, in which we were able to obtain, or we unfroze some serum that we had from the year 2000. We had a stock of serums from the year 2000. And the number of serums is the second line of the table. And the first virus to which that was faced through the marginalization was the virus that we isolated CU PH1N1 associated to the pandemic, then the human virus, and then the swine virus, and the last column the swine virus of the type O and up to. The thing is that if we had isolated in 2009 and we had been in contact with pigs previously, and you can see how in the year 2000, 28% of the serums came out positive to that virus that is associated to the pandemic, and 6% and so forth. So we corroborated that this virus associated to the pandemic had already been circulating among the population of pigs, backyard pigs in the city of Mexico. The next one, please. Also, another one of the findings that we have started looking for in this first stage was a subtype H1N1 that is identified as Querétaro, Mexico 35, that was isolated in 2010, that had incorporated the gene of the polymerase A coming from a subtype H3N2. In other words, a recombination in one virus that is the H1N1 that we have reported and that is in the gene bank. Please, the next one. To this moment, we have two viruses of the type H1N1 circulating among the pork community in Mexico. One of them that went from the avian, from the water bird to the humans and then to the pig that we are finding now. And the other subtype, H1N1, that came from 2018, it went on to the pig directly, and it has remained within the population, the swine population for many years. In polymerase A, as I mentioned, coming from a subtype H3N2 from the human beings. In this first stage, till 2014, we had two viruses, H1N1, and the same type, but with specific mutations and specific recombinations in a subtype H3N2. The next one, please. In regard to the type, subtype H3N2, the origin was aviar and 3 of its genes, and we keep it in the neurominase, and that goes from the water bird to the human in 1998, and then from the other genes from the bird to the this was the virus that we had up until 2014. That's the virus we had. For the next stage, please. Next slide, please. And what we were finding, or what we were looking for, it was to look for the greatest variability at the level of hemagglutinin and neuraminidase, but also in some of the genes of the isolated virus. Next one, please. We found in some of the states of the Republic, the previous one, please. We found the subtype H1N1 and H3N2 in the state of Jalisco, then H1N1 in the state of Michoacán. We find in two states, Mexico and Guanajuato, the subtype H5N2, which is a subtype that affects the birds. The subtype H3N2, and then later H1N1 and subtype H2 and 2 in the state of Guanajuato. In other words, that we're going to see more description of this next slide. The next one, please. If you see this first phylogenetic tree of the phynotenine, we go to the tree, please. Yes, okay. We have in the phylogenetic tree a sample from Guanajuato and another one from Jalisco. We have serum isolated in 2015, and we go further down to an independent cluster, Guanajuato also, from 2015. In gray, we can observe an H1N2 that had been reported in Canada and the United States, but not so in Mexico. And then after that, a subtype of H1N1, also, but in another different cluster to those that we have described before. Two orange virus that are found in porks, but of avian origin also. And two more virus that are subtype H3 and 2. We go to the next one, please. The neuramide has a pattern of phylogeny that is very similar. And the most important thing here is to see how we distribute the different genes compared to their origin. In other words, species and origin, as well as based on their viral subtype to which they come from. The next one, please. Here we have a table of summary that also comes in the report that is written and distributed by Procinort. It turns out that the first virus that is in gray, the swine, the subtype H1N2 is not the same subtype that is reported in the United States. It has 3 genes that are equal. The subtype that is reported in the United States for H1N2 is insulin is a protein of matrix, but it has nucleopeptide that come from a subtype H1N1 from the pork, and there's a gene that is neuramidase that comes from the subtype H3N2. I repeat, it is not the same subtype H1N1 that is reported in the United States. There's another one that is called subtypes in porks. Literally, only the low plasticity pork have it. Then we have 2 virus, H1N1. And the first from Jalisco is only, do you have, and we have the subtypes, and it's H1N1, that is a traditional subtype of porks. Then we have the one of Guanajuato. Everything is for swine, but except for one that is not a H3N2. And then we have the third subtype, H1N1, is of human origin. We have found a virus, H1N1, in which all their genes correspond to the human strain. And the last 2 are subtypes H2 and 3, H3N2, sorry. But nevertheless, you can see that the last one is a completely swine of the same origin subtype, and that one before last is from Hidalgo presents on the one hand the 4 genes that come from that subtype H3N2, and the other 4 genes that are incorporated from a subtype of the origin H1N1. Next one, please. It is published the complete genome sequence of the subtype H1N2, because precisely it is different than the virus that had already been reported in the United States and Canada. And there is an emphasis to the relevance that the monitoring of a new viral might have in Mexico. But apart from that, with different things that they have reported already in the United States and in Canada. The stakeholders, at least in the country, are the combination of the species. Some of the cases, the porks also eat some chicken meats, possibly infected or not, and then we begin with a interspecies transmission. Here we have a photograph in the bottom that I was talking. This was taken by a student a few years back in a sampling in Chiapas, if I'm not mistaken, and there we have 2 species coexisting. That is a factor that is a predisposing of the interspecies transmission. The next one, please. Also, as Dr. Tavis had mentioned it, we have made a 3D modeling see if those changes of amino acids have implications or not on the antigenicity of the virus, or maybe even because of the polarity of the molecule. As you can see, where you can see there where we have a green circle comparing the one on the left to the one on the right. We have more blue and red, that is with a positive and negative polarity because of those amino acids that change in that zone. And the one on the right, it has a zone that is more whitish. It is neutral. In other words, what we try to do always is that to see the lineage, because it is important to do a translation to amino acids, and then later identify if there are changes in the protein that might imply a greater pathogenicity or more facility of transmission to other species. The next one, please. What in reality is matters here, but at the same time is something that is concerning, is that this is a map from 2019 in which we have identified the most prevalent subtypes of influenza. It is reported by the WHO. Then you can barely see on that side, on the side of pie, that the color, the blue color, very light blue color, is talking about the presence of the subtype H1N1 pandemic. And then it goes on with a gray color that talks about influenza and subtype 1. And then we have a green color of influenza, subtype number 3. And then you see another color that is a little more intensive in green, which says influenza A and not subtypified. I remember that a little while ago, Dr. Long put a slide that says that our lab receives thousands of samples that we sequence to identify the pathogens. Are they all identified or no? What does this mean? That we have a great area of opportunity to be able to detect, at least, according to what we see in these graphs, what the viral subtypes are being located, for example, there in the United States and Canada. The greatest graph of the country, or the majority of the country is influenza type A, but not subtypified. And this is where we have to go to between Canada, the United States, and Mexico to have the capability of sequencing and, of course, the responsibility of reporting what it is that we have in our countries. And I close by commenting that in the year 2001, in China, we found a virus or a sequence of the subtype H1N1. From 2011 and 2018, that sequence reappeared prevalently in swine. They call it H1N1 G4. And in the last 4 years, it's already in humans. They consider it with a great pandemic type of a risk. So it is a call to attention for everybody that is involved in this, the constant monitoring and report of our subtypes. It doesn't matter the species. In this case, it demonstrates that there are viruses that have been able to reach the interspecies virus. The influenza virus has very partial words, but also recombinations among other viruses of other species. And of course, the relevance that there are viruses that are passing around in porks directly of human origin or with recommendations that have allowed it to adapt to the pig. That is all on my side. Thank you very much.
Muchísimas gracias, doctor Iván.
Thank you very much, Doctor. I'm sure that there's a lot of questions, but we're going to continue on with our next presentation. This is by Dr. Jorge Lindo from Icata. Welcome, Doctor.
Sí, buenas tardes. Bueno, yo les
Well, good afternoon. I'm going to present to you some of the preliminary results that we have had in a project that is being executed
en el CENIT, aquí en
in
Palo
the
Alto.
Senate here in Palo Alto,
Bueno, estamos trabajando
Palo Alto, where we are working.
localizadamente en el estado de Jalisco, ¿no?
We're working very localized in the state of Jalisco.
Bueno, el
Well,
doctor Iván
Dr. Ivan has already given us a small, given us a very good explanation regarding the virus, what it is, and so forth. And the segmented virus, the next one, please, where we can, where we have process for reordinance or genetic reorganization that gives that way to new subtypes. The next one, please. I'm going to move very quickly on this slides to talk more about what we're doing in the state. And well, this is something that also can give way to a great variety of clades within a subtype that also have an effect. And like Dr. Ivan has already mentioned it, it has a lot of effects in regard to the infectious process, and also regarding the immunity that could be generated. The next one, please. And well, this is the characteristics that break out the garments of the illness. As we can see, well, we can have up to 198 combinations because of the amount of amino acids that have been reported. And nevertheless, within that, we also have clades that can generate a problem. Next one, please. This slide is one that we have also seen in which the receptors that are in the cells of each one of the species, well, they play a very important role in the interspecies infection. Next one, please. And also talking a little bit about what it is, the infection of influenza and the of the swine fever. And this is also something that opens up the possibility of other illnesses or of characteristics that will help also in providing the susceptibility to the narcotics. The different production units that exist in the place and this is important to see when we go out and do business, and you're going to see why it's important in the state of Jalisco. Well, as everybody knows, the women that can request petitions and abortion and so forth, and when we, in the men, we have the affection of the spermatic presence and everything, and this comes to have an effect on the production. And well, for the adequate control of the influenza, there are many factors that can be taken into account. We have the biosecurity, the management, vaccination, and especially the diagnose, which is a very important part of the. We precisely need to know what type of subtypes we have, and where does they belong? Where do they belong? So I will end up doing the high and establishing which are the ones that are going to be established. And following it as populational medicine from the epidemiological point of view, it is important to understand in what regions what is circulating so that we can even refresh our diagnosis system. Next one, please. Well, this is the protocol that I was mentioning. It is a protocol that is being held. It is a project that is being carried out by the NIFAB and it's centered in the state of Jalisco. The state of Jalisco for several years now has been working on a project which has implied diagnosis of several illnesses and the job with different organizations where we have had to come to have results that are very important, especially talking about or learning to know the types of production systems that we have in it. That map that we have there shows different regions in which we are working on. And the lighter color corresponds to a region that has a low density, animal density, while the rest, a higher environment corresponds to a density of approximately 160 animals per square kilometer. While the other one we have 93 animals per square meter. And this is another factor also in the presentation of the illnesses. The objective population are swine farms of low and small scale in the state. And well, to tell you a little bit about this, why in Jalisco? Jalisco is the main producer of pork meat. It produces 23% of the total production, the national production in Jalisco. You have 279 production units. This is data of the National Association of Swine Producers of the state where the majority of this the areas are places for work. And we are talking about 1,632 units of scale for low production, while there are 1,117 commercial farms. So this makes us have a great variety in regard to how the system is operating or the management of production in each one of those places. So, the next one, please. We are trying to focus on these problems and talking about the swine fever is one of the main viral illnesses that affects the economy of the farm. Its distribution is very, very important in public health, and we have been registering losses associated to the daily gain of weight, mortality, waste, pigs, higher consumption of food, and so forth. And this has been calculated approximately. It is 70 pesos per hog, and when there are co-infections with other viruses, it can be up to one. It can come to up to 100 per dog. And added to other parameters that can be quantified, and we can say that we have up to a 5% in the increase of the cost of production. So there's also a very broad development of technology, biological and biochemical. Jalisco has characteristics that are very particular to it. Of the production, it is necessary to adapt technologies that are attached to what is circulating at that moment. The next one, the next slide, please. In such a way that the objective is to carry out an epidemiological study of this virus in the state that will allow to have the development and the application of technologies in it. Within the objectives, the specific objectives, well, it is the identification of the farms of a large and small, low and small scale, where we can do the diagnosis of this virus to detect it, isolate it, and this is also going to be a part of the study, but doing the study will be logic, and that will allow to identify the genetic types and the distribution of this virus. Sorry, the next one, please. Well, in Mexico, well, the biggest challenges for the influenza are the prevention and the control. Yeah, we have identified a level in the swine production with data that I presented to you before, and we have some being activated. Logically, well, the fact of being able to have updated diagnosis systems based on the genotypes that are being circulated, well, this can also come to generate an impact within the regions for which this type of system might be adapted. The next one. The next one. This project considers the sampling that we might come to even sample 99 farms and tell them that here the sampling is by convenience and trying not to look for to find something if we to find the virus. Therefore, to this day, we have been able to sample 13 farms with clinical background of the respiratory system related to the virus. And, well, we have obtained also 33 pools of five samples of flower fluid, and we have obtained 168 soil samples that were taken individually. Within those those years of change. This 10, 12, and 20 weeks. Next one, please. For the viral identification, well, we carried out an extraction of the NDA. We also carried out PCR, real-time PCR, and justifying also the GenM, the positive samples with a set less than 30 that were sent to be subtypified and to sequence through the supervision of Sanger, and the analysis of that was done. And we were able to see this in the subtype H1 subtype 2, and we were able to have this comparisons. And we also carried out serological sampling. We did a test for inhibition of the hemagglutination, and this was done for H1 and 2 and 1 and H3 and 2. Next one, please. And the results. And well, now what we have to this date in the project, well, we found 10 the samples that were taken out of the 13. There were 5 nasal swabs of the 33 that represent 15%. 2 positive farms were also identified as positive through the samples of fluid fires, and that are and the biggest distribution of the positive forms was between the one of 3 and 6 weeks of age. Next one, please. The positive samples. Well, here in my case, the positive samples are expanded for the segments 4 and 6 of the hemagglutinina and neuraglipidasa. And these expanded total with 6 of the 7, where we obtained the adequate concentration to be purified and eventually be able to sequence them. At the end, we found 6 sequences. At the end, we got 6 samples. The one was from the oral fluid, and others were from swabbing, and the sequencing allowed us to identify 2 subtypes mainly, the H3N1 and the H3N2, which is the slide that follows, please. Thank you. Here in this table, well, we have the sequences that we obtained by subtype, and in pink, the ones that are in pink are the ones that subtypes that come from sporting origin, and the blue ones come from human strains. These are of human origin. Well, in this case, H3N1. There we find H3N2, and let me tell you that to this moment we have only done the characterization of molecular transformation, but we're about to do the isolation so that we can do the reporting for it. Yes. With regard to the receiving of antibody for the subtype of H1N1, well, the majority of subtypes were found between week 12 and week 18, in which, well, we found peaks also in the week 9 and the week 21. In other words, where we found the positive titles, majority of the positive titles, and this corresponds to the weeks in which the pigs are in the week number 9 of age. The highest subtype was between weeks 6 and 9. That is shown there with a bracket. And we also had high titles, and the highest peaks that were also located in the week number 9, and then has a blue label marking it. For example, the level of participation is the lowest coefficient of variation that we had. And within that week, the hogs, the pigs that were sampled that week. And the next one, please. Well, these data preliminary data that, as I say, we still have samples that we have to process, and we are working with a regional union of pork producers and the protection association of the state of Jalisco through the epidemiological survey department that are helping us in identifying the farms that do some sort of to carry out some notice of problems, breeding problems, so that we can go in and do the sampling. And this has been a process very slow. It has not been as massive sample taking because at the end of the day, what we want is to make it up the amount of sample to find the virus in this case. And well, the data that we have up to now, it demonstrates the circulation of influenza in the state, and that is something that is well known. And the circulation of different subtypes associated to the sorry, a little while ago I said H1 and 2. That reported correctly. I refer that it had not been reported. It's H1 and 1 because we only have the molecular characterization and we don't have the isolation. So this is something that also reflects the need of doing a more exhaustive studies to be able to identify other subtypes. And as mentioned previously by the previous panel members, to come to the identification of the clear and to see what is the dynamics of infection, or also in the immunological part of the state also. Well, also to associate and be able to associate the presentation of the subtype with some characteristics, pathological characteristics, and as mentioned, what is the immune response that is being developed. And the next slide, please.
Identificado casos positivos, se han realizado seroperfiles.
As a conclusion, then we have found some profiles, and in a generated way, it's preliminary. We continue to work in the development of this project, obtaining samples. The next one, please. Next slide. And that's the improvements we have in this project, the steps forward. Also to let you know that the state group of epidemiology surveillance and the AB, they're doing national samples of federal resources for influenza. In this case, they do so in the trace level, where they see the lungs. There's 13 traces that are being sampled in the entire state, and every month we go and take the necessary samples from it. They haven't detected any positive influenza samples in the year, and then for the amount of samples that we have, the number of farms that we are trying to find the virus, right? And how we are able to do a diagnostics and a typification at the end of the day that give us information so that we can afterwards be able to make this technology sample samples, and we are able to refresh the diagnostic kits that can be generated. And that's what we had. Dr. Francisco Rivera Benitez is the person in charge of the project. He's in charge of this project. And any questions that you may have, you can contact us in these emails. The first email is mine, and the second one is Dr. Francisco, whom is our primary. Thank you, everybody, for your attention.
Muchísimas gracias, doctor.
Thank you very much, doctor. As we mentioned before, we have one last presentation that was not part of your agenda because it was added last minute, but this presentation is by Roxanne Motroni. I hope I pronounce your last name well. She is a leader for a task force in the United States. Dr. Roxanne, can you hear us?
I can. Can you hear me? Okay, great. I'm going to share my slides. Can you all see that?
Yes, we can see it. Just make it bigger. Yeah. Well, no, that's in the mode of, now it's ready.
That's right. Okay. Thank you, everyone. I'm sorry that I can't be there today in person, but it's my pleasure to provide an update on what the USDA has been doing around highly pathogenic avian influenza. So what I'm going to talk about today is the research locations that have been carrying out this work, what the ARS response has been to the outbreak in poultry, what our outbreak response is in livestock, and then also what our planned research, and share some preliminary results as well. So we're very fortunate with the USDA. We have 2 high containment laboratories with the expertise and the facilities to deal with this high path avian influenza outbreak. Of course, there's the U.S. National Poultry Research Center in Athens, Georgia, that has been studying high path avian influenza since the 1960s, and is the reference center for avian influenza, as well as a world collaborating center. Their specialized facilities allow them to work with a variety of poultry. And over the years, they've not only worked with high path avian influenza, Newcastle disease, but also coronaviruses and West Nile virus. The second facility that has been doing a lot of the response to this is the National Animal Disease Center at Ames, Iowa. And they also opened in the 1960s to study diseases of economic importance to livestock. And because they have high containment BSL-3 facilities, they've been able to respond to a lot of our emerging diseases in livestock species, like with swine influenza, SARS-CoV-2, and now with high path avian influenza. And I think as well as Tavis pointed out, they're a collaborating center with the NIH through the Centers of Excellence for Influenza Research and Response, which has enabled them to respond very rapidly to this current situation. So just a quick refresh, in February of 2022, there was a new strain of high path avian influenza detected in the US. And then as of June 2024, it has been detected in 48 states in commercial or backyard flocks, and has affected about 96 million birds. The ARS response to the current outbreak in poultry, we've done a lot of pathogenesis studies to understand how this virus infects and transmits in chickens, turkeys, and ducks. The center also updated and validated diagnostic tests to make sure that the NALM, or the National Animal Health Laboratory Network, and APHIS were able to detect the virus. They also have done extensive testing of licensed diagnostics and vaccines to ensure that they work against this current virus. And as David talked about today, they supported vaccination studies in California condors using vaccines. And then they also updated that vaccine that was developed in 2015 to be more closely matched with the current circulating strain. So I think the future research plans, or the continued research plans for the U.S. National Poultry Research Center, is to continue testing vaccines and different vaccination protocols in turkeys. That will start in July. One of the big questions is whether or not the vaccines that we have will work in turkeys, given that they live longer than some chickens. We'll also be looking at additional infection and transmission studies with the mammalian isolates, and also doing more vaccine studies to ensure that they work against the current circulating viruses. When we switch to thinking about high path avian influenza in livestock, in March of 2024, this was first detected in livestock in a goat kid in Minnesota, where there were poultry that had been depopulated, and the goat and the poultry shared a water source. Then shortly thereafter, HPAI was detected in dairy cows in farms in Texas and in Kansas. And in April, the 1st of April, the first person within the United States tested positive for the H5N1. And this was the first reported cow to human spread of H5N1. And then there was a second and a third case reported in May. The first 2 cases were primarily conjunctivitis. Whereas the third case, the person was actually reported some respiratory issues as well and was started on antivirals. So then in April 2024, USDA APHIS announced a federal order requiring testing prior to movement of any lactating dairy cattle and mandatory reporting of influenza. I think as David talked about in his talk, it was understood that primarily the spread of this disease was mainly due to cattle movement. And we, in May of 2024, ARS researchers, specifically Tavis and others, used phylogenetics to determine that the current outbreak occurred from a single spillover from wild birds into cattle in December 2023, and then sort of slowly circulated until it was diagnosed in March. And so the federal order was really put in place to try to minimize any future spread of the disease, and has worked fairly well. But that brings us to today, where it's currently in 12 different states, with Minnesota, Iowa, and Wyoming being the latest states to have identified infected herds. I also want to draw attention to the USDA APHIS HPAI website. This has all of the latest information in terms of the affected herds, and this is updated every day about 4 p.m. Eastern time, so that you can see this information in as real time as possible. So after we heard about all of this, about the dairy cattle issues, USDA ARS kind of jumped into action. And so this is a timeline of the work that we have done primarily in support of this. So once the virus was detected in dairy cattle in March 25th, USNPRC started pasteurization studies using benchtop pasteurization methods, and using milk as spiked milk samples, because that was obviously the first question, is whether or not our standard pasteurization protocols really inactivate the virus. And so I think, it goes without saying, or it's really important to note that the same day that this was detected, USDA ARS started pasteurization studies, mainly because we have the expertise in the facilities to do so. This further evolved throughout April to testing bulk milk tank samples as well as retail milk samples, and eventually ended with, or not ended, but is continuing to show that pasteurization studies or pasteurization is effective in inactivating the virus, and that that, as David presented, that a small portion of retail milk products contained viral RNA. As well, I think as David alluded to in his talk, we have moved to bringing a continuous flow pasteurizer into containment at the National Poultry Research Center to be able to test pasteurization as it truly occurs in the field, since doing it at the benchtop, there can be a lot of different errors introduced. And so those studies are in the process of are still ongoing and being done in collaboration with the Food and Drug Administration. We also started testing immediately on pen-side tests, so to be able to facilitate looking at, how can we continue to move milk, how can we contain continuity of business, how can we rapidly test. So David did a lot of those studies as it relates to lateral flow devices. I think David also touched on the fact that then the question became, is meat safe? And so USNPRC, in collaboration with the Eastern Russell Research Center, did some studies using a low-path AI provided by USNPRC to ERC, where they spiked ground beef into patties, cooked the patties, and then showed that under normal cooking conditions, so long as you don't eat your hamburger rare, the virus is inactivated. And if you leave it at 120 degrees or a very rare burger, there is still decrease in viral load, but it doesn't continue or get rid of all of it. Also, at the same time that all of these milk studies were going on at USMPRC, we started moving into trying to figure out what is happening in the cattle. And again, because of the longstanding expertise there in working in influenzas with Tavis and Dr. Amy Baker and Dr. Bailey Aruda, as well as the high containment facilities that can handle large animals and the ability to get dairy cattle, we were able to sort of rapidly move into doing infection studies to try to get a handle on how are cows getting infected, how much milk are they, how much virus are they shedding. So really about a month after the first diagnosis in Texas dairy cattle, cattle studies began at the National Animal Disease Center. And 2 routes of challenge were tested. The first were in some heifers. They did a respiratory through nebulization challenge study. And then in 2 lactating cows, they did intramammary challenge. The respiratory inoculated cattle didn't really show too many clinical signs, but I do believe they did seroconvert, whereas the intramammary challenge cattle did show signs consistent with what they were seeing in the field in terms of depression, fever, drop in milk production, all those things. So those studies, like I said, they started the end of April. They went into sort of the end of May. And so those samples are still being analyzed, and Amy and Bailey are busy writing that paper, but that should hopefully be available soon. At the same time that all of this was happening, Tavis and his group was working with APHIS to look at the molecular epidemiology using all of the sequencing and the tools that he's built through the swine influenza program to show again that it really seemed like it was a single spillover event that was then transferred through cattle movement. We've also been looking at, or there's a real push from our leadership and others to see whether or not there are vaccines available that can be used in some of our livestock species. And so we've started having conversations with our own Office of Tech Transfer, as well as APHIS Center for Veterinary Biologics, to see what other commercial companies have vaccines out there for us to consider testing in case we actually need to vaccinate cattle. So lots of work has been going on in the last 6 weeks, and it's really quite incredible that Dr. Suarez and Dr. Anderson were here today to present on it as well. So I greatly thank them for all of that work, because it's been a crazy 6 weeks trying to get a handle on what all is going on and making sure that the food supply is safe. So again, preliminary results, the WIN paper is out that shows that there is likely a single spillover event from wild birds to cattle in December. We are working on developing a challenge model and an infection model in order to be able to potentially move into vaccine development or to test vaccines should we need that. Again, these are very long, complicated studies, particularly with large lactating cattle in a BSL-3 containment. And as you, I'm sure, are well aware, when you necropsy these animals, there's a lot of samples and there's a lot of sample analysis that's still ongoing, but hopefully have this information published in the next month or so. And we're also planning our second cattle study starting kind of mid-July. And then again, obviously, the food safety considerations David talked a little bit about lateral flow diagnostic assays, so ways in which we can test bulk milk samples or have pen-side tests available for cattle. Completion of the commercial continuous flow pasteurizer work, and also repeating that. And then as he also alluded to, testing of the retail dairy products provided by FDA. And if there's interest in that, that is published by Erica Speckman. And so all of this work, like I said, has really taken place in, I think, the last 6 to 8 weeks, and was done heavily requested by other agencies such as USDA APHIS, the Food Safety Inspection Service, the Foreign Ag Service. The Condor work was done in support of the Fish and Wildlife Service, as well as the Milk and Public Health and Food Safety was done for the Food and Drug Administration, as well as CDC. As well, in all of this, we recognize that our own BSL-3 space is limited, so we have been working with the Research Alliance for Veterinary Science and Biodefense, or the the RAVEN network, to maximize the amount of BSL-3 space available to be able to conduct those studies. So with that, I'm happy to take any questions, but also wanted to make everybody aware again of the USDA APHIS high-path avian influenza website that is updated daily for real-time updates, as well as the CDC HPAI website. And that's all I have. Thank you.
Thanks, Dr. Roxanne. That was very helpful. I'm sure there are a lot of questions. Sorry, I'm going to change to Spanish. Vamos a tener
Okay, so we are now going to have some options to have your questions for the speakers. And I don't know if the speakers that are here in the room can want to come back to the front, or you want to stay in the back. We can have a mic available for you to answer any questions that are presented to you. We would also like to that after the questions, the objective that we have discussed here within the leaders of the different teams is to try to go into 3 or 4 specific items that are important to follow up on this sort of activity, right? 6 points, and then may find gaps in the knowledge that are fundamentals, and that we can focus our resources and our time. And then I don't know if there's any questions here in the room. And just to break the ice, I'm going to start myself with the questions. We have a few questions here for Dr. Betancourt and for Dr. Barbosa. From Dr. Aruna, he wants to know what is the most common sample used to do an porcine influenza in the swine influenza in Mexico. I said nasal or pleural fluids. I don't know who wants to answer that. Nasal swabbing or pleural fluids.
Gracias. Bueno,
Thank you. Well, in our case, what we use when we were looking for traces, we wanted lungs. And the entire first stage of the research was lung samples from swines that have been harvested already. I remember that out of the 1,000 samples, only 24 were positive. The second stage of the research was more targeted. These were nasal swabbing in positive farms for weaning swine. And so we were more successful in the second stage. But at the beginning, we did lung samples for tracing, and we did nasal swabbing on weaning work in the second stage. Thank you. Now going a little bit more into detail on the question here, the people from Animal Health here can help us answer this. There's an epidemiology surveillance that is done routinely in Mexico for this type of disease. So I like to think that the most common sample to support is the nasal swabbing. All right, is that correct? Yes, the answer is yes. The nasal swabbing is the most common sample taken for active surveillance and in swine. Yes, that's just correct. And it can also be done through traces monitoring as well. And in animals that were identified as sick, we take lung samples from them, and that can be sent to the lab. In the case of the passive surveillance, yes, it was ideal to send the lung in as together with some samples from pork that was alongside the animal that was sick, so that we can do comparisons.
Thank you. How often the oral fluid samples are done? Is it done at all in Mexico?
Nosotros no
Well, we don't use cotton. It has to be a swab, a Dacron swab, and it has to be suspended on PVC so that we can extract genetic material.
Tenemos otra pregunta.
We have another question from Dr. Aruna. We also commented to our participants in Zoom. If you have any comments or questions, you can write it down in the chat, and our team here will let us know what the questions are. We have a question from for Dr. Aruna and Dr. Roxanne. The question is if all the cases of influenza in bovine cattle is related with animal movements only.
Did I understand the question correctly? Is the transmission only due to animal movement?
Yes.
So I don't think that's fully known at this point. Right, I think that's the working hypothesis. But certainly I don't think it, you know, as we have more outbreaks, I don't think we can say that that's the only thing that's happening. Tavis, I don't know if you wanted to answer anything, if you have anything updated since your last paper to share.
No, I think, Roxanne, that's correct. The initial spillover and then the initial epidemiology was a single wild bird origin into dairy cattle, and then pre-symptomatic or asymptomatic dairy cattle movement spread it. Once it gets into the farming premise, the mode of transmission remains unknown, and that would be the active studies that are being conducted by USDA ARS that Dr. Matroni covered, trying to work out how it's moving once it's on the farm. Thank you.
Right, and there's also a question in the chat of what was the reason for intramammary challenge, and I think to carry on with that, you know, we don't know or didn't know if the cows were infected, you know, from a respiratory route as you would expect with an influenza virus, or if it was happening more in the milking parlor as you move from one cow to the next with the milking equipment. So those were why we chose those two challenge routes to see if we could recreate the disease.
Thank you. ¿Tienen alguna otra pregunta aquí en el foro?
Anyone has a question here in the room? Any other questions? Okay, so now maybe I have a question for Dr. Travis. During your presentation, you mentioned the reordering of the virus and the implications that this may have on developing vaccines to try to avoid a pandemic jumping into humans. But my question is more towards what can we do to prevent this from spreading in our swine so that we can avoid it more or less getting into humans? How do you know? Do you have any activities that you're taking in that fact to try to have a vaccine for the swine part?
Right. That's a good question. I think in general, we see nearly 100 different genome patterns circulating in our pigs in the US. And there's a subset of those different gene combinations with increased transmission efficiency. But we've only experimentally validated a tiny fraction of those. So we have an idea that reassortment is very important, but we're not yet able to model exactly what different gene combinations will result in a particular virus increasing in detection frequency. So for a vaccine perspective, the approach has been to conduct on-farm surveillance, either passively or actively. And ideally, you get a bit of a mixture of both, with the passive surveillance that happens year-round, and then targeted surveillance at particular points of the year or in hotspots and different interfaces. And using that genomic surveillance data, we're able to work out what the dominant genetic types are within specific regions or locations. These days, a number of the biologic companies in the US have licensed vaccine platforms. So then they're able to take those specific genetic sequences and drop them into this platform and then deploy that for vaccines. And that tends to be the general approach. That most of the farms are vaccinating sows. Sometimes the little weaned piglets are also getting vaccinated now as well. And that would be a vaccine targeting the hemagglutinin gene. And then sometimes it's the neuraminidase gene as well. And then matching it based on genomic surveillance. Ideally, that approach then minimizes the circulation of those specific viruses. And if you minimize diversity, ideally, you also minimize the opportunity for reassortment, and you minimize the probability that one of those viruses gets into people. So, yeah. And a lot of the research we're doing is trying to optimize what you put into the vaccine and what are the best components, what are the best combinations, and then what are the best vaccine platforms to use. Thank you for the question.
Perfect. Thank you. Perdón. Muchas gracias.
Gracias. Muchas gracias. Thank you very much. And with that in mind, and given the emergency we have with avian flu in bovines, have you considered vaccination in cattle to try to contain the infection as well?
No sé alguien que me pueda contestar de los panelistas.
Any of the members that would like to answer that? Any of the panel members that would like to answer that?
So, the decision to vaccinate cattle is being discussed within USDA, but there are a lot of trade issues with that and a lot of issues around, if we're vaccinating cattle and not vaccinating poultry, those kinds of things. But we have been tasked with being prepared and trying to develop a vaccine that we think will work in the event that the secretary and others make the decision that they want to vaccinate cattle.
Okay. Muchas gracias. Tenemos otra pregunta.
Thank you. We have another question in the chat. We were asked, I don't know, it's not addressed to anybody in particular, but we have several experts in influenza here that can answer it. They want to know if epidemiology surveillance in wastewaters at swine farms, it's really feasible. Maybe Dr. Betancourt can answer that. Thank you. Well, remember that there is a lipid layer in the virus they obtain from the cell at the moment of the exotosis. So in theory, we could, without a problem, and in fact, we don't test in wetlands with migrating birds, we are able to detect influenza virus in wastewater from swine farms. I guess we could, yes. Now, but we have to take into consideration that in the case of pigs compared to the birds, and the virus is not in the gut, it's in the respiratory tract. And so, keeping that in mind, we could look to detect the virus in wastewater from the swine farms.
We have an additional question to the panel. If the panel could comment on the predicted effectiveness of avian vaccines given viral dynamics in the avian prairie pothole, for example.
I can start to comment on that. I sort of alluded to in my talk that surveillance in regions like the prairie pothole region can have some predictive power as to which genotypes are going to be popular in the Southern Hemisphere and during overwintering seasons. So if vaccination in those regions were to become an effective strategy, surveillance in the prairie pothole region could help predict which strains are coming, although the situation is not quite as complicated as the one Tavis has to deal with multiple antigenic shifts in different H1 and NAs. So, but there could be antigenic shifts, or sorry, drift, that having some predictive power might be beneficial, but I can't, vaccine development isn't necessarily my purview, so I can't really comment on their predictive effectiveness at this point.
Ok, muchas gracias. Creo que no tenemos más preguntas en el chat.
Thank you. I believe that's all the questions in the chat. Or do we, Dr. Anderson, you wanted to say something?
I think Dr. Suarez presented earlier with some nice data on the California condor, showing that as long as they got seroconversion in those specific birds, they had relatively good protection, and that's derived from 20 odd years of study. So I think that perfect matching between what's in the field and what's in the vaccine may be less important in some subtypes, but it still requires active surveillance to track and then the studies to determine when there is a reduction in cross-reactivity. Yeah. So I don't know if David Suarez is still on. I think he may have dropped out, but hopefully I captured one of his major points. Thank you.
Thank you, Dr. Anderson. Me parece que ya no hay más
Thank you,
preguntas.
Dr. Anderson. I believe there's no more questions in the chat. I don't know if anyone else here in the room would like to make a question. So if there's no more questions, I would like to ask everyone here in the room to help me summarize 3 ideas that were takeaways that we can have from today. Now, what do you think is the priority on the steps ahead for to continue to research this type of interaction? Let's go to Dr. Rosalena first. Thank you. Well, congratulations to everybody. I believe it's been a day of a lot of information, very productive information. I had a lot of answered questions, but also I have a lot of other things that come to mind. And with that in mind, and answering your proposal, you know, some of those takeaways would be, or concerns would be the variation, the constant variation of the virus and its capacity to evade or to be detected, you know, and what Dr. Anthony mentioned, and how can we make sure that these different variants can be detected and they are not, you know, being ignored by our diagnostics. That doctor already mentioned about H5, the H1 and the H3, right? How and what can we do? Yeah, that's the question, right? What can we do to improve and ensure that nothing is falling in between the cracks? And another situation along that same line is the surveillance for vaccination, right? You know, how important is it to continue the epidemiology surveillance? And we have more challenges piling on, not only water birds, land birds, swines, and but now we are adding bovine to it, so that creates the more difficult scene, and we need more people collaborating to move forward on this thing. And then last, I would say, is that my personal interest will be to be able to identify what was said by Dr. Davis was about the different virulent capacity of this virus. Aside from this multi-basic region, what other region could be involved or connected with this interspecies jump, and they could, in fact, be able to reach the human and be able to have a human-to-human transfer. Those were my 3 takeaways: vaccination, diagnosis, and surveillance.
Muchísimas gracias, doctora. Tenemos por allá una manita levantada.
Okay, I see a hand raised also. I agree completely with the idea that we have heard a series of very good presentations regarding different topics related to the surveillance, related to the diagnose, related to the way in which the image, virus of the influenza is affecting the different species and how it is swiping from one species to another. I think that we are facing a great puzzle with many pieces that are together, but then that are not put in order. And I consider that it would be necessary to start strengthening something that we have been listening to in the last maybe 8 or 10 years, and that we don't see very much it's crystallized here. We have specialists from all sizes and colors and shapes and flavors here in the room, and nobody has said how do we do it to manage it under the criteria of one single health. So I believe that it is a very correct and ideal moment to start building through the ICA, through the agencies, through the ORISA, through the offices of the countries, clear structures as to how to interact with criteria of one single health to attend to a problem that is definitely affecting all of us. Thank you. Thank you. The truth is that this goes very much in hand with what Luisa just mentioned, the interinstitutional cooperation. In Mexico, Canada, and the United States, we have the experience. We have groups that have been put together for a long time. There are national plans, there are response plans for the attention of emergencies. So I believe that it is just to bring those connections closer. I wouldn't like to talk anymore about this. Thank you. Yes, in fact, I believe that you're right. We need to bring our international interinstitutional cooperations closer, precisely to go deeper into this topics and the aspects of one single health. Because we saw recently with the news, the case of the person that passed away that had, or that was identified of a virus H5N2, I think it was. It seems to me. And there is a series of misunderstandings in the media that in the beginning they created, if you want to see it, sort of like a red flag. They're calling the attention that this was going to be the beginning of another pandemic, and it's the truth. The virus of the influenza has this type of strength. They are pandemic viruses, and that's why it has us here together. So we have to issue a message of alert that does not scare the community, but we have to be very careful. But with regard to that, we have to give a very clear message of what has to be done to prevent the infections, and in this case, identify it and control it. And we'll avoid all the repercussions that this might have with the economy of the country, like we saw with the virus of SARS-CoV-2. And I think that that would be one of the key points that would have to be followed. We have another comment here by our secretary, Dan Chow. Thank you. To tell you that there is a good news because the ICA is leading the One Single Health. We had a summit in September last year, a continental summit, not only in the north, all the member countries participated, and it was an opportunity for agricultural health, and we continue on cooperating with them, and it is us a mechanism of cooperation. It is different that talks in that program, but as a secretariat, I'm going to invite you so that you can establish the bridge, that you can see that there is all this effort going on beyond this room. There are more people. Get to know more people, more researchers, regulators, people from the private sector. Those.
Gracias. Complementando
Thank you. And complementing in the sense of what is being commented by all the members of what was commented by Susana and those before me. As a matter of fact, we were talking over lunch of that importance of having a strategy for communication of adequate dissemination with all the scientific vigorness, so that when these type of events take place or situations or something, well, immediately the science, the academia, the institutions that have then mandate or that support of the science to effectively be able to provide a message, a clear message that it's not about hiding things, but to put them in the adequate dimension, and that the population, that society effectively, it might be well informed. We were talking about this great problem, the infodemia that we have right now of the fake news. So I think that this is a great opportunity. These platforms, these ecosystems of research, that you as researchers, as scientists, as technicians, that are supported by all that experience, that this might be something that might be that might use a strategy in the matter of awareness and communication and dissemination in a recurrent manner, not every 3 months, not every 6 months. It would be something that it would have to be done more than necessary. And complementing the part of one single health at the ICA, we are promoting all this concept, but not anymore just as that, as a concept, the case of success. And as a matter of fact, I invite you and I tell you that we are going to celebrate the second Congress, International Congress of One Single Health in the framework of what is an initiative that is co-organized with the National Agricultural and Livestock Council and the CENICICA, the ICA, and other institutions. And that is going to be carried out on the 5th and 6th of August. And apart from that, we're going to have a parallel event organized by ICA, in which we can also invite you, if there's not at least virtually, that you can accompany us, and where we pretend that we continue on disseminating beyond the concepts of a case of success, and something that is so relevant to public health, the human health, the animal health, the environmental health, the health of the soils, etc. Thank you very much. Thank you very much. I don't know if there's any other comment from our panel members that might want to help us to summarize everything that we have just seen, a conclusion on your side.
I'll let Roxanne try
May
and say something.
Thanks.
No, I think one of the earlier people in the audience wrapped it up together quite nicely. That frequently we all have very different backgrounds, and so getting in the room together and talking about genomic surveillance, analytical techniques, and how that it can improve animal health is probably one of the most important components. But it requires long-term investment where there isn't competition for resources. There's just the view that we're trying to improve animal health and human health, and that if we work together, we can achieve a lot more than we could independently. My background's in evolutionary biology and fish, and now I'm working with pigs and viruses, and it takes all different types, and you have to be open to that to ideally improve and enhance and achieve that end goal. So there we go, to Roxanne. Thanks.
Yeah. I think to piggyback off of what Dr. Anderson said, I think what we've seen in this outbreak and in previous outbreaks is the need to have these long-term collaborations and these long-standing relationships in order to move quickly to address the questions that are most pressing. And that, I think that it's really, really difficult. It's really difficult within the US to coordinate the research. And so I think doing it internationally when you have different, different agencies with different missions and different funding sources, it becomes infinitely more complicated. But that as we try to address these really difficult questions, there is the need to bring in all these different perspectives and all of these different expertise to address the question, because it's not just the genetics questions or the genomics questions or the pathogenesis studies. There's a whole part of this that is epidemiologic. There's a whole part of this that is environmental. What is climate change bringing to it? And so there is this need for the coordination, and the longstanding relationships, I think, to be able to address the questions that are before us. So that's sort of what I took away from this.
Muchas gracias, doctor Roxanne. Y creo que tiene razón. Es importante tener un equipo.
Thank you very much, doctor. we agree that it is necessary to have a multidisciplinary team to be able to fight all these illnesses. Now I give the floor to Dr. Betancourt. Thank you. Well, in reality, I would just like to comment that independently of the relevance of the institutions in Mexico and in other countries, I believe that the access should be focused on emerging illnesses, in this case, today, the influenza. And if we have as a country, Mexico, or if we have in Canada, the United States, or a working team to attend to this, I think that it would be very important to institutionalize and structure the links, formal diplomatic links for the exchange of knowledge and information that is taking place day by day, day after day. At least I perceive that this forum is a great opportunity to be able to establish links. At least we in the faculty of veterinarian medicine, there is a research team on birds and wild birds of influence, let's say, a group of research in pork or swine, and a group of research in wildlife. And I believe that, and I'm sure that we are willing to get together and to contribute any type of information that is required. And I refer to in the binding aspect with one single access. In other words, interinstitutionally, focused on one health and always with institutional respect that is required. There is a lot of information, not only the one that we have today, that I believe could help between the three countries to be able to create working access. This is a very severe illness, and what is happening with the H1N5 and bovine, or what we can expect with the A1 and 2 with this subtype N1, G4 that is already present in humans in China. I think that it is a moment to do something. Okay, thank you very much. Okay, we have one more comment with Dr. Barbosa. Yes, thank you. Well, going a little more in depth in what Dr. Ivan is saying, well, we from the part of the INFAP, we have a very close relationship with the producers at the field level. And well, what you say about that companionship on behalf of other countries, that those diplomatic relationships that will IMIFAP would be more than happy to try to participate and to be there from the point of view of the producer. And well, this is something that also expands the characteristics of the presence of this illness, and also to lend the information that is generated, so that at the end of the day, in addition to one single health, we might also have a joint effect on the production and the life quality of the producers. That at the end of the day, in our case, and the INDIFAP, they are a very important part that we attend to. That would be all on my part. Well, okay, with that, thank you very much. The truth is, I'm very happy to have all of you. I have specialists from different areas and different institutions and from different countries, because this has been something that has been very enriching for those present here and those of us who are participating through the Zoom conference. We take back all these notes, and please be sure that we will follow up to this as it is custom of ProCNorth and of this working force on animal health. I thank you enormously to all of our panel members, the participations, to the guests, and well, we expect you tomorrow once again at 9 o'clock in the morning for another day of work. Thank you all. Have a very good evening.